It is difficult to use the host infection stage of T. cruzi in experiments because amastigote is an obligate intracellular parasite. Our culturing technique allows the amastigote to temporarily replicate outside of the host cell, so we can perform experiments directly on this clinically relevant stage of the parasite. Demonstrating the procedure will be Yukie Akutsu, a technician from our institute.
Maintain a host parasite co-culture according to the manuscript directions. When the Cas9 expressing parasite is ready for differentiation, collect the supernatant into a conical tube and check for sample quality under a microscope. If there is a significant number of extracellular amastigotes, perform a swim-out procedure to isolate the trypamastigotes. Spin down the mixture of trypamastigotes and amastigotes for 15 minutes at 2100 times G. Then, discard most of the supernatant, leaving 0.5 to 1 milliliter of medium in the tube. Loosely cap the tube and incubate the pellet at 37 degrees Celsius for 1 to 2 hours, which will allow the active trypamastigotes to swim out of the pellet. After the incubation, transfer the supernatant with the trypamastigotes to a 1.5 milliliter microcentrifuge tube. Spin it down and resuspend the pellet with 5 milliliters of DMEM. Transfer the parasite to a T25 culture flask and incubate the flask at 37 degrees Celsius under 5% carbon dioxide in a humidified incubator. Around 95% of the parasites will differentiate into amastigotes after 24 hours. Centrifuge the culture of extracellular